Okay, hi everyone. Sorry for a few minutes late, but I had to connect everything and I had to be sure that everything was working. So, on the event of today, we're talking about the IMAT and how to ace the IMAT test in 1 month. As you can might have
Okay,
seen.
hi everyone. Sorry for a few minutes late, but I had to.
Sorry, but I have my voice double. Anyway, as you might have seen and have heard, the decree now is out, so we have a few information more that we're going to give you today. So, for the guys that probably don't know us, this is us. We are student for students. So basically, we are all med student or medical field student. As you can see, we have different shirts because those shirts are the one of the subject of the test. You know, you have four different subjects in the test: logical reasoning, biology, chemistry, and maths and physics. And with me today, there are four students that I will introduce, actually three students that I will introduce in a few minutes. So what do we usually do? We do free university orientation, such as the event that we're doing today. We give advice on IMAT based on our experience, because although probably not all of us have studied or are studying in an IMS faculty, so basically we are not all of us are studying in English, although every one of us has actually took the test back in the day, so we know what you have to do and how you guys are dealing with the old procedure right now. We do have one-to-one personal coaching to help you to design your study plan and get as ready as possible to the test. We do have online intensive live courses, and we usually are covering both theory and practice part. We do have exercise book, both physical and uh, digital ebooks. And for the international student of you, we can provide you the ebook as well. You can find everything on our website. I will give you the links and all the information to find us online as well. We do have online simulation and online support that are free and available for everyone that needs help for anything, and online simulation free as well. And we do have summer school in a project that actually started this year in August in Milan, where we both are doing a meet between the culture of the student from Italy and from Milan specifically, and and then in you can meet all the people around the world that are taking these summer school. It's a very nice experience, and you can find all the information on our website. So, what are we doing today? We start with a quick IMAT update because, as I told you, the decree is finally out, so we have to keep in mind some dates, and we have to clarify some information. Then we'll take in, you actually are taking a simulation, an IMAT simulation, and we are going to correct it. So be with us, stay with us until the end of the event, even because I will give you a little gift if you are staying for the whole event. So with me, by the way, I haven't introduced myself. I'm Riccardo Mingarelli, and I'm starting. Actually, I just finished the fifth year of med school, and I'm currently in the sixth year of med school. I study in Sacco Hospital, which is in Milan, and with me, you have other teachers and three girls, wonderful girls that can introduce themselves if they want to.
Yeah, hi everyone. I'm Geraldina Guiducci. I also study in Milan. I study in the IMS program, which is medicine in English. I'm at the fourth year, which is almost done, so I will start the fifth year soon. So yeah, also if at the end you have any questions specifically regarding Milan, feel free to ask. We will have some time also for it. And I will be your math and physics teacher for today.
Hi, I'm Sara and I study in Pavia. I'm actually starting the 6th year. If you have any question about Pavia and anything, just feel free to ask. And today I'll be introducing logic.
Hi everyone, I'm Madeleine and I study in Turin. I'm at the end of my 2nd year, so almost in the 3rd year. And today I'm going to be teaching chemistry.
Okay, sorry, but I need to check if someone, because they told me that they don't see us on YouTube. I want to make sure that they do. Wait. Okay, they told me that they see me. So mm, if for the one of you guys that are on YouTube and they are following us, please type on the chat so I can actually see if you if you can follow us. Just try to type on the chat if anyone else see us. Okay, I hope that people can see us actually, because they just told me um that actually, because someone says that probably there's a problem with the link, I don't know, of YouTube. Probably the live it's different, but actually I don't know how it is possible. I don't know. Let's go ahead. So, the ministerial decree. So, just wait a second. So, because I just saw that people send a new YouTube link. So I wait just a second so that all the people can get into the new link, which I don't know why it's different though. Okay, now I see the live. Okay. Now I see. All right. So, perfect. So there were some of you guys that couldn't see us, some of the other could see us. So sorry for the one that were waiting for us, but unfortunately there was a problem with YouTube. So, anyways, not many important thing we were saying. We were just introducing us. So, I'm Ricardo, by the way, and I have three other girls with me that are going to be your tutor for each subject: logical reasoning, chemistry, and maths and physics, and I am the tutor for biology. So, what's important though? The ministerial decree is finally out. A few days ago went out. So, we now have the important dates that you have to keep in mind. So, take a screenshot of this slide and please listen careful. What? So basically, from September 2nd to September the 12th, and it's important the 3 p.m. Due hour, because after that time, you can no longer apply for the IMAT. The IMAT registrations are going to be open. So when everything is, when September 2nd, you want to register to the website, you have to go on doing all the procedure both on university and the university and of the center where you're taking the test. So if you have any doubts on this information, we did have an event, and I will take talking about that in a few moments. But for the moment, just keep in mind September the 2nd, September the 12th. Then on September the 17th, you probably know that already, you have the IMAT test, and around 10 days after the test, the anonymous rankings are going to be out. The anonymous ranking are the ranking list of every person that took the test, the IMAT test, and got all the requirements to enroll to the ranking list. And basically it's anonymous because you only have a code with numbers and letters. Then on October the 3rd, you can finally see your personal scores on University page, on your profile on University, which is a website for doing to do all these procedure on. And then you have on October the 10th, finally, the national ranking, where you will know if you finally are assigned to your first choice university, or if you have to wait until the scrollings to enroll in your first choices universities. This is also important because you have the this is important because you have the centers where you can take the test. There are two information that you have to keep in mind: that if you are an EU student, or you're taking a test in Italy, you have and you're taking a test in Italy, you have to keep in mind that you have to go and enroll in your first choice university to take the test. So it's not, you cannot choose the closer university to you. If you want to apply for the Università degli Studi di Milano, so Milan University, you have to take the test in Milan University. On the other hand, if you want to take the test in any other state where you can have an IMAT centers, you can find all the information about that on the website. If you have any problem, you can try to contact us so we can try to figure it out with you. Unfortunately, we do not have all the information about every single state, so it's important that you try to do that with the information of your state. And in any case, you can try to contact us in our customer support. Then, as I was saying, if you have any doubts on how to do the procedure and all the other things that you might want to know, you can go on our Med School Test Buster account on YouTube, and you can see that we did have another event a few days ago on all these kind of informations. If you don't have time to watch everything, I just want to underline that you do have a simulation and a correction that is a free exercise, so it might be important for you and might be useful to go and see at least this information, and of course the information about the program, the IMAT program, and how to register to the university, and all the information that you have to access to the IMAT. And if you want to, or you prefer to read this information, you can even find us on the website, on our website. We do have a blog where we wrote some articles about every single thing that you have to know, both let's say technical things, for example, how to what you have to know about the syllabus, so each topics that will get into the test, or all the information that you have to know to get to the test ready as well. But we do have also information about, for example, each university where you can study in Italy, and other information that might be interesting for you. So scan the QR code that you find here, and you will find all the information that you might need. So now I want to open another page, or if you want to use another device as well, it's fine. Just type imatsimulator.testbusters.it. We do suggest to use as a browser Chrome, Google Chrome, because it performs better usually. You can use the email that you use to register to this event, or if you want to, you can create another account as well. Just keep in mind that after you put and you insert the email, you will get a code, a 6-digit code, and you might find it on the spam. So just double check. If you don't get the email immediately, just try to check if you have it in the spam. So I will wait just a couple of minutes so you can do all this procedure, and I will show you in the next slide where you find the simulation that we are going to correct and doing together today. So just type me on the chat of YouTube if you have any problem, if you can log in and everything. So go on the simulator and go on the available simulation that you have, and specifically on free IMAT simulation, as you can see here. Once you go on free IMAT simulation, you have to look up for the simulation prep school 2024, which will be the simulation that we are going to do today. So I still wait for you guys on the chat to tell me if you have any problem, or if you could find the actual simulation. And whenever of you guys are there, I will start the timer. I've seen that someone of you guys asked, when do the classes usually start in the public units? Well, it depends on the university itself. Usually, as the IMAT, the test is usually pretty late in the fall. You might start around November. It depends on the university, though. So whenever all the decree of the university is out, you can read every single things, and the universities usually tell you when they are going to start with lessons. Usually for IMS, it's not going to be very early in the fall. They are waiting a lot because, of course, as you were seeing, um the ranking list and everything is pretty late in October, so you might have some time before then starting the lessons. Okay, so I've seen that people are in the simulation, so see you in 100 minutes. We'll be back here. I'll start the timer, although the simulator as well has the timer. See you in 100 minutes, and we will start with the correction. All right, we are back in. I'm sorry for the awful sound that the timer has. Anyway, so for you guys now, I'm asking you a very important thing. If you scan the QR code here, the QR code has a link for an important evaluation questionnaire that we ask you to fill because it's an important feedback that we have and help us to improve this kind of event. So it only takes 2 minutes. Really, it's very, very quick. It's very fast. So I'll let the QR code for another couple of seconds and then we'll move on and I will ask you again. And please do that because in that way we can give you the little gift that I was talking before about, that is at the end of the presentation. So let's move on to the next few things that I have to tell you before then start into the correction of the simulation that you just took. So we would ask you to follow us on Instagram on any page that we have so that you can stay up to date until the date of the test and even more after that. We do have plenty of information on Instagram. We do have a lot of stories that explains you things and that help you to stay updated. For example, the day of whenever the decree was out, we did a couple of stories where we were explaining everything. We do have some evidence stories like here where we do have some mnemonics and other tips for you guys. We do have posts with questions and other important thing that you might want to know, and it's a very nice way to get those information without opening a website and whatever. We do have our website, which is medschool.it. There is the QR code once again. And we do have a lot of information on here. For example, the link to the online simulator where you can find other free available simulations and even simulation that if you want to buy to exercise yourself, you can go on there and you find all the information about that. We do have university guides that can help you find your university, the university that you most like. For example, whenever you have to move to Italy, and all the information that you have to know about once again the decree, how to do the process to enroll to the university and whatever. We also have IMAT live courses, an online live course with the theory part from the 19th to the 25th of August, and the practice and revision part, which is between the 5th and the 15th of September. It is not going to be 3 weeks long, but it's just the weeks that is going to cover during the lesson that we're taking. We do have a complete test busters IMAT question bank that you can find on the simulator with more than 1,300 quizzes. We do have a video library with the explanation of some certain important and the major topics that you'll find in the test. And you can also add a personal coach. And all these little piece of skills that you can buy, you can also have it whenever you buy the live course that we have. You will get the quizzes, the question bank, and the video library as well. And the personal coach, as I was saying at the beginning of everything, helps you making your study plan in order to get as ready as possible to the test. We do also have an essential theory book, as I was saying at the beginning of the event today. We do have it on physics. We do have an ebook that you can buy. We do have physics books as well, but unfortunately we do not ship them all around the world. But you can buy the ebooks so that you can use it whenever you want, and you have theory, formulas, and all the information that you have to know, that the essential information for the test. And we do also have practical exercises with the solution that are explained so that you can easily understand what the mistake might be, and even they are important because from there you can get the theory back refreshed if you want to. So let's start with the explained solution. I will hand the speech to the girl from logic, because I am not allowed to explain logic because it's not my subject. So go ahead.
Okay. Says you have to end the screen recording. Okay. Mm-mm. Okay, can you see my screen? Perfect.
Yes.
Okay, then we start with the logical reasoning part. If you have any questions or comment, you can feel free to chat in the YouTube chat, and we'll
Oh
make
yeah,
sure
I'm
we answer
sorry.
your
I'll
questions.
just interrupt you to say that, guys, unfortunately, we do have a little delay because we are presenting on Zoom, and then you guys are watching us on YouTube, so we have a few seconds of delay. So using the chat for us might be a little complex whenever we are explaining. So if you have any question, any doubt, you can write that on chat, and we will answer you back whenever we finish explaining, or the other of us might try to explain that thing for you if you didn't get a word. Sorry again.
Okay. So then we start with the first question from logical reasoning part. We were presented with 3 scales, and the question was which type of candy should we add in the place of the question mark, so here, in order for the scale to balance. Let me know if you can also see my pointer. Can you see it?
Yes.
Yes, okay. So first of all, I noticed that the first scale is very much similar to the last one. So in the first part to the left, we have a square and a circle. And in the second part, we have a square and a triangle. And here again, a square and a triangle. It's only missing one square. And what I noticed is that the first scale is in equilibrium, okay? So basically, it was very easy. We only had to compare the first and the third balance and see that they were quite equal, and then we could understand that it was only missing one square. So the right answer was 1D, D that says a square candy on the right plate and no candy added on the right plate. Okay. Then second question. This one was about a deposit. So we were talking about Paul deposit. He had a first initial deposit of $1,500. And the text said that the bank every year added a 5% to his initial total of 1,500 every year. So for the first year, we had to add a 500. And then for the second year, we had to add a 500 on top of the final amount of the first year. Then the text says that at the end of the 2 years on which the money stayed in the bank, Paul withdraws only a half of the money. And with part of this withdrawal, he buys some t-shirts for that cost $155. And the final answer is how much money does he have left over buying the t-shirt. So what do we need to do is to calculate 2 consecutive percentages. The first year, we start with a total amount of 1,500, and we have to add 5%. So first we calculate the 5% of this amount, which is 75, and we sum up 75 to 1,500, and we get to 1,575. Then we start with the second result, which is the final amount of the money he has in the bank after 1 year has passed. And we again add a 5%, but 5% of this second amount, so 5% of 1,575, which ends up being $79. So we need to add $79 to 1,575, which finally makes 1,654. Then the text says that he withdraws half of the amount he has earned in 2 years, so half of this number, which ends up being 827. But we also know that with 827, he spent some of this money in t-shirt, exactly 155. So finally, we just need to subtract 155 from 827, and we get to the final solution, which is basically how much money he has left after buying the t-shirts, which is $672. Okay. Then we move to the third question, which was about syllogism. And it was asking which of the following is necessarily true. Pay attention to necessarily. So let's start with the first one. Every x is y. So we might draw the x circle inside the y because every x has to be contained inside the y circle. Then it also saying some z are y. Okay, so this might or might not encompass also the x's. We're not sure. But what does it say? Then no z is x. We cannot know that because we know that the z is intersecting the y, but we cannot know for sure if it's intersecting or not intersecting the x. So the first syllogism is false. What about the second one? No y is x. So the two circles must be drawn, one not touching the other. It's also saying no z is y. So the z is not touching the y, but the z could potentially intersect the x. We are not sure, okay? What is the final statement of the syllogism? No z is x. Actually, this cannot be said. What we can only say necessarily true is that the x doesn't touch the y, but we were not given precise information about the z. So we cannot know if the z is a completely independent circle or if it might, on the other hand, touch the x. So this is also considered false because it's not considered necessarily true. And same goes for the last one. Some x are y, so they might intersect. Some z are y, okay? Then some z are x. It might be possible, but it's not necessarily true because the z could also be found not intersecting the x, okay? So when they ask you for something necessarily true, it's something that has to be written in the text. Otherwise, it's not necessarily true. So the final solution was that all the three were wrong. And so the answer was D, no one. Okay, then we move to number 4. This was a straightforward question. So basically, we were asked for the definition of a substituted asset, which is an economic mean, okay? And the only right option for the right meaning of a substituted asset was B, so 4B. Basically, a substituted asset is a good or product that can be used to replace other identical goods. And it could be helpful to look at the name because to substitute means to use something instead of something else, which then has to substitute its job, basically. So they have to be identical. So 4B. Another straightforward question was number 5. We were asked about Denmark and which of the following country does not have the same form of government as Denmark. So first we needed to understand that Denmark is a constitutional monarchy and United Kingdom is the only one which is a parliamentary monarchy. So it was the only wrong option. Sorry, so 5 was E, the right answer. Then we move to number 6. So this question was talking actually about penguins' poop. In particular, Adelie penguins, which are penguins from Antarctic coast. Their poop is pink. And what actually, first of all, this was a long text. So first we go and see what is the question, final question. The final question was which of the following is the best expression of the main conclusion? So we needed to find the main conclusion of the text. So in a few words, the text was saying that as this penguin, they make a bright pink poop. This is used by scientists to identify their colonies and study them even from space, because the pink bright color can be seen from space. So what is the main conclusion? I would say something very similar to A. Adelaide's penguin poop can be scientifically used to identify their whereabouts, okay? The other ones were not main conclusions. So B, for example, penguin poop is colored because of their diet. I mean, it is true, but it's not the main conclusion of the text, while the other two, C and D, were wrong, totally wrong based on the text. So 6 was A. Then we have a mathematical problem. So number 7, we're talking about two people, Claudia and Fabio. The text was saying that they are meeting each other at the train station, and Fabio is running at 21.6 kilometers per hour, while Claudia only 0.8 meter per second. Be careful, because here the units don't match. So we either need to transform Fabio velocity in meters per second or Claudia into kilometers per hour. They are divided by a space of 34 meters. So first, two people are running towards each other. We convert because it's easier, Fabio's velocity. So from kilometers per hour, divide by 3.6, and we get to 6 meters per second. So now we calculate the velocity total, which is a sum, because the two people are running towards each other, so they might meet sooner, and that's why we need to add up the velocities. So 6 meters per second, velocity of Fabio, plus 0.8 meter per second, which is the velocity of Claudia. We get a total velocity of 6.8. So we have the velocity, we know the space, we calculate time. Time is 5 seconds. But then if we go into the answers, we don't find any answers saying 5 seconds. So probably we need to convert 5 seconds into hours. And what do? We divide twice by 60. Okay, so seconds divided by 60, we get the minutes. And then we divide this number by 60 again, and we get the final answer in hour, which corresponds to letter C. Okay, so 7C. Then we had a huge text talking about people's tendency to form friendship. And what the text was basically saying is that we make friends only with the people with the similar odor, okay? So that was the main thesis. And the question was, which one of the following, between all of the options we had, would most weaken the above argument? So the argument is that we only make friendship with people that we can smell and that have a similar smell to us, okay? So the right answer was actually E, because E was saying that it was shown by research that people with anosmia people are people that cannot smell any smell, still tend to be friends with people with the same similar body odor. So if this is right, then basically smell is not the real determinant of friendship, because if they cannot smell and still they're friends with the same people, then smelling is not the let's say, what drives friendships. So the only option that weakened the argument was actually E. So number 8 was E. Then we moved to the last logical reasoning question. This was a graph, okay? So it was talking about 4 teams, team 1, 2, 3, and 4, competing in period 1, which was basically a sum of different physical games, and period 2, which was mental games. So first, let's say a competition of physical and then one of mental. The question was, there were 4 possible answers, and which one was correctly inferred from the graph. Okay, so we have to start from the first one and basically rule them out one at a time. We could see just by looking at the graph that the A, C, and E were wrong. For example, A was saying team 3 scored the lowest in both challenges, while, for example, if we looked at the period 2 challenge, the lowest scoring team was team 2, not team 3. So A was wrong. C was wrong because it says that the minimum score in the physical test was scored by team 3, and actually in the physical score, the lowest score was scored by team 2. And then E, team 3 scored the last amount of point in the mental state, while in the mental state, it was not team 3, but team 2 who scored the lowest points, okay? So then we have the 2 missing question, which are the other one, because they are saying that the total points of team 2 are equivalent to those of team 3 subtracted by team 4. In this case, we need to do calculations. So we need to sum up this value and this value to get the final points scored by team 1, same for team 2, same for team 3, and same for team 4. And then we need to compare them. So team 2 is roughly 62.5, team 3 is 50, and team 4 is 100. So what was option D saying? That the total points of team 2, so here 50, are equivalent to those of team 3 subtracted those of team 4. And we can clearly see that this is not possible. So option D is wrong. Basically, we're left with the final option, which is option B, which is correct, in fact, because it was saying that team 1 scored the points of team 2, so team 2, 62.5, added to team 3. which makes up perfectly 112.5. So final 9B was the correct option. Okay, I think we're done with logic. And now if you have any question, I'll be ready to answer on the chat.
All right, let's move on to biology. So I share the screen. Okay. So the first question of biology was about the digestion of certain macromolecules. And what you guys have to keep in mind is that sometimes the name of the enzymes might tell us what they are going to do. The correct option was B, because lipids are digested by the lipase, and this takes place both in the small intestine and in the mouth. You have to keep in mind too that the option, for example, C was partially correct, because in fact we do have the digestion of proteins in the duodenum, but it actually is completed by the trypsin, which is another enzyme, because the pepsin, which was the one on the option C, it's the enzyme that we can find inside our stomach. So remember that lipids and starch, and so the carbohydrates starts the digestion in the mouth, and then it proceeds in the small intestine, while the proteins start the digestion directly in the stomach. So this is something you have to keep in mind. Next question was about the brown adipose tissue, which is an important tissue that has the function to produce heat with the thermogenesis, which is a process where we have the decoupling of the phosphorylative oxidation in the mitochondria, so that we do produce heat. In fact, the correct option was the. We can find these kind of tissues around those essential organs, such as the heart or the lungs, for example. And usually we found more of this tissue in the kids, in the babies, rather than in adults, where we usually find the white adipose tissue. Let's move on to the next question, which was about immunology. It was asking about the myeloid cells function. You guys have to keep in mind that lymphoid lineage cells are the B, the T cells, and the NK cells, which are the natural killer. The B and the T are part of the adaptive immune response, while the NK are usually part of the innate response. While the myeloid lineage cells, we find many different kind of cells, like the granulocytes. The correct option of this question is that we can divide once again in neutrophils, basophils, and eosinophils, and these are important because they were the option of the correct option of the question, and because they are help us against parasites infection or bacteria infestation, and the allergic reaction, and the release of histamine and other proteins that can help us fighting certain organisms. So the correct option was C. The 13 was about the enzymes that charge the amino acid on the tRNA. This has a specific name, which is aminoacyl tRNA synthetase. Once again, we can try to figure the name out thanks to the name of the enzymes. Of course, you might have excluded the helicase or the primase or other enzymes that you might know that they are part of other process. In this case, the aminoacyl tRNA synthetase is the one that actually catalyzes the charge, the esterification of the amino acid onto the tRNA, the specific tRNA with a specific anticodon on its arms. So let's try to say that when you find this kind of answers and this kind of questions, you might have to reason on and think about the name of the enzyme, so you can try to work the problem out better. Let's move on to the 14th. I'm sorry if I look down because I'm reading the question with you guys. So here it was asking where basically we can find the glycogen, which is the form where the glucose is stored and stocked inside our body. We have to remember that we have two organs, the liver and muscles. Muscles are more selfish, let's say, because the glycogen usually in a physiological situation is given only to muscles. So muscle stock the glycogen and gives that to the muscle itself, while liver is more generous because it's stored all the glycogen and gives that to the whole body. That's because basically muscles needs a lot of glucose in order to function, so they want to keep that inside them. By the way, whenever you have a certain pathologies, you might have the destruction of muscles in order to get the glycogen from them too. So the correct options was E, the liver. And you have to keep in mind that whenever glycogen is given from the liver to the bloodstream, we have the increase of the glycemia, which is the value of the glucose inside our body. Glycemia means glucose in the blood. Let's move on to the 15th. Here we had a little tricky question where we have to know the stops and starting codons, which are AUG, UAA, UGA, and UAG, which are the ones shown in the slide. And from there, we had to find the anticodons, which are the one that we can find on the tRNA. Remember that a codon is a triplet of bases, of nitrogen bases that we find in the mRNA, while the anticodons are these triplets that we find in the tRNAs, as you can see from the picture. So from here, we only had to find the counterpart of each triplet. So we found that the correct option was D. So option 1, 2, and option 4. The 16th was about biotechnologies, and it was asking when we understood that the genetic material was made of DNA. We have to remember that Griffith discovered that there is something that transforms cells, and so he discovered the transforming factors, the so-known transforming factor. Then Avery tried to understand what was this transforming factor, and he found out an hypothesis that the transforming factor was actually DNA. But you have to remember that at the beginning of these kind of studies, scientists didn't really like DNA because they thought it was a very simple molecule, so they couldn't realize that all the genetic material, which was so complex and so important, was made by such as tiny and very easy molecule. So they were thinking that it was actually protein, the base of our lives. But then Hershey Chase, they confirmed that it was in fact DNA, let's say the alphabet of the genetic material. Mendelssohn was the one that discovered that the DNA replicates in a semi-conservative way, and then of course Mendel is the father of the modern genetic, or better, the Mendelian genetics, so the one that you guys might already know. So the correct option was C. The 17th was about the let's say the macromolecules in general, and it was asking what statement was not correct. We have to remember that we have different kind of bonds that links the different molecules that forms a nucleoside and a nucleotide. We have to remember that a nucleoside is formed by a sugar that can be ribose or desoxyribose, depending on the macromolecule itself. the ribose in the RNA and the oxyribose in the DNA. And the difference is in the carbon 2, so the C2. The option that actually was the correct option of the question, so the option B, said that we have the bond in the C2, but it's wrong, because at this level, what we actually see is that sometimes we might have the oxygen and some other times we might not have the oxygen. So it cannot create a bond. Probably the chemistry teacher might explain that better for you guys, but basically the oxygen is essential it's crucial to form this kind of linkage. So without the oxygen, you could not build the nitrogenous base on the sugar. So the glycosidic linkage is at the level of the C1, so the first carbon of the sugar. Let's move on to the 18th. Here, the question was about what kind of microorganism or organism has the RNA might have an RNA as a genetic material. You have to keep in mind that any organism actually has a DNA as a genetic material. So protozoa do not have RNA as genetic materials. They do produce RNA whenever they need to translate the DNA into proteins, but they do not have the information in form of RNA, such as the metazoa, the bacteria, the protists, and animal cells as well. The only exceptions are viruses, which can have any kind of genetic materials you have in mind. They may have double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA. They have the majority variety of genetic materials because they are not actually microorganisms. They are parasite. They are cellular parasites, so they need the cells in order to survive. Otherwise, they could not survive, and they could not share and spread all around the world. So yeah, this is the option. The correct option was D, so viruses. Let's move on to the 19th, and as I said before, if you have any question, you can type us and ask us on the chat on YouTube. So during the mitosis here was asking what following structure are divided into the two daughter cells. And you do not have to consider the cytokinesis. This information was crucial to answer the question properly, because you have to remember that mitosis, it's the division of the nucleus. Okay, so the nucleus divided into two nuclei, and so chromosomes and the nucleus, of course, are going to be the only options that divided during mitosis. All the others organelles and all the other structure are divided equally. They are not actually divided like in half. They are not split apart. We give some of the organelles to one cells and some of the others to the other cells during the cytodeaeresis, so the division of the cells itself. Okay, so the correct option was A. So as you can see from the slide, the organelles are evenly distributed during the cytodeaeresis. And remember that mitosis and meiosis are division of the nucleus only. The division of the cells that might happen or might not take place in the cytodeaeresis. So the 20th question was about the function of the third complex. This question specifically was a little tricky because you had to remember how many H plus were pumped by this complex. You have to remember that this complex takes the electrons from the complex Q, the molecule Q, which is the ubiquinone. So it reduces the ubiquinone in it. It oxidizes the ubiquinone in ubiquinol, so an alcohol, but it also pumps 4H plus into the intermembrane space. So this is very important. One of the options said 2H plus, and so that was the wrong option. And you have to remember that then after that it gives the electrons to another complex, which is the cytochrome C, which is this little knob that you can find here in the picture. Okay, so correct option was letter D. The 21, it was asking for each molecule of glucose, how many ATPs are obtained from Krebs cycle and the oxidation after Krebs cycle. Here, once again, you have to keep in mind what the question is about and what it asks you. So it asks you to per each molecule of glucose, which is an essential information, otherwise you might miss the correct option. So we have to remember that each Krebs cycle produces one ATP directly from GTP that actually is formed here, then 3 NADH and 1 FADH. Then we have to remember that from 1 NADH we get like 2.5 ATP that we can round up to 9 ATP, and from 1 FADH we get 1.5 ATP that we can round up to 2 ATPs basically. So here we have 3 NADH, so we have multiplied the value per 9, and then we get the 12 ATPs in general that we get from the oxidative phosphorylation and the Krebs cycle itself. From here, we have to remember that one Krebs cycle is, or better, that from one molecule of glucose, we undergoes into 2 different Krebs cycle because we have 2 acetyl-CoA that are generated per each molecule of glucose. So we have to multiply everything times 2, and we have the final option that was 24 ATP, or option C. Let's move on to the 22. Here we have to remember each phase of the meiosis. So here are all the phases and what happens during this kind of phases, and it was asking where we basically find the chiasmata. The chiasmata, as you can see here, are in the diplotene, which is one of the final part of the prophase 1. Remember, these 5 phases are in the prophase 1 of meiosis. So during meiosis 1, at the beginning, we have this subdivision of the prophase because something important happens, such as the crossing over during the Pachythene, which is another phase. And then we have the chiasmata, which are basically the proof that the crossing over took place during the Pachythene. Okay, so the correct option was in fact E. So none of the above were, none of the other options were correct. And you have to remember these phases because these are very important. I don't know if you have any mnemonics that you want to share with the other audience, or if you have any kind of mnemonics. I did have one in Italian, but as yesterday I went out watching the stars because you have, you know, the falling stars. I did find another mnemonics that you might find helpful, which is let Zeus punch Demetra and Diana, which are the 2 goddesses from Greek and Latin word. So if you wanna, you can use that, or you can find yourself. So just remember that these phases are crucial and very important because usually you might find many question on this during the exercise that you're taking and in the test. And if you wanna, I can repeat the mnemonics, which is Let, Zeus, Punch, Demetra, and Diana. Let's move on to the 23rd, which was what of these statements about the Krebs cycle is correct. The Krebs cycle, basically we call it an amphibolic process because both catabolic and anabolic reaction and products take place. So basically what that means, it means that during Krebs cycle, we are using molecules, so we undergoes in catabolic processes, but we do also have the reaction and the build-up of other molecules, so it's anabolic as well. And for this reason, we can call it amphibolic, so both catabolic and anabolic process. So the correct option was the letter B. The 24th was what of the following statements about retroviruses is correct? You have to remember that retroviruses are viruses with a particularity, which is the reverse transcriptase, which is an enzyme that can convert the RNA into DNA once the virus infects the cells. So, for example, the HIV virus, when it goes into the lymphocyte T, for example, it starts generating the DNA by using this reverse transcriptase, okay? So it integrates the RNA inside the DNA of the cell. And it creates these DNA, we call it all coding with the little C before than the DNA term, because it doesn't have any kind of introns, it only has exons. So basically we call it all coding DNA. And so the correct option was option B. So they use reverse transcriptase to copy RNA from their genome and inject it into the host cell or the infected cell. So the 25th, which of this molecule can be found in a plant membrane? Once again, the terms and the questions are very important in here. So the plasma membrane of a plant, it's basically just like the animal ones, the eukaryotic ones. So it's composed of phospholipid bilayer as the other membranes that you know, and it is semi-permeable. The cell wall, on the other hand, it's composed of cellulose, which was one of the option A. But remember that cell wall is something different from the plasma membrane, which it's something crucial. Otherwise, the cell doesn't exist. We cannot define a cell without the plasma membrane. So the correct option was none of the above. So letter E. The 26th. Which of the following option about point mutation is correct? In here, we had to remember that a point mutation is a mutation that affects only one or two nitrogenous bases. So it's a very short mutation that can have different kind of effect. We can have a silent mutation if the codon that has changed is the same code. It's basically a codon for the same amino acid, because you know we might have many amino acids that are codified by different codons. So if we have this kind of mutation, basically nothing happens. It can be a missense mutation. So we basically generates a different amino acid. So we do have an important effect because we have a different proteins at the end of the day. We might have a nonsense mutation where we change one or two nitrogen bases, and we generate a stop codon, so it generates a shorter protein, and so it generates a very huge effect because the protein, it doesn't work anyway. And the most dramatic is the frame shift, where we have an insertion or a deletion of one or two nucleotides that can change the frame of the of translation of all the other codons and all the other amino acid after the mutation. So this is the dramatic, because if it takes place at the beginning of the process, well, here we have a completely different structure with completely different meaning, and this is very dangerous for us. So the correct option was C, where we were defining the nonsense mutation. In the 27, we have a question on the sympathetic system, nervous system. So basically, this is it's part of the peripheral nervous system, the autonomous one. So the part that we do not control, that controls the involuntary reaction to certain things. And we have to remember that the sympathetic and the parasympathetic has different kind of reaction. In the first case, in the sympathetic system, we do have something called the fight or flight reaction, while in the parasympathetic we have the rest and digest reaction. So basically, in the first one, it's something that helped us during our past ancestors' lives whenever we were fighting in nature. Helped us to increase, for example, the heart rate, to had more bloodstream going through the muscles and run faster. For example, it does have the mydriasis, so the increase of the diameter of the pupil, so that more lights get inside our eyes, and we can see better. We can see we can see even more farther than what we would see, and so we could escape from a dangerous situation more easily. Then we do have a vasoconstriction, so a reduction on the volume of the blood vessel in the peripheral blood vessel. We do have something in the respiratory system. We do have a bronchi dilatation, so basically the increase of the volume of the bronchi. This is why, for example, if some of you guys have allergies and got an anaphylactic shock, got the adrenaline or epinephrine as a medication to increase the heart rate, so the heart could pump, because it was a shock, so the heart wasn't pumping enough. And the epinephrine also dilates the bronchi, so you can inspire and get the air through your lungs, otherwise you won't. An important thing is that in the male reproductive system, we do have the ejaculation, which is part of the sympathetic system, while the erection, it's something that undergoes the reaction of the parasympathetic system. So you need to be relaxed in order to get the erection, basically. So correct option was E. The 28th, we in this picture, in this question, we did have a picture, and it asked us what part of the stomach we had with the letters, and the uppermost part, so the one that you see here, it's actually the fundus, so it's basically the uppermost part, and then we have the biggest part of the stomach, which is the body, and then we have the antrum, which is the one that connects the stomach to the duodenum here, so to the small intestine. Remember that we do have two sphincters, which are the cardias, it's the uppermost one, and then the pylorus, which is the one that connects to the small intestine. The functional part is because the cardias is not a sphincter, because we do not have muscles that close the sphincters, but it's a functional one because we do have certain kind of actually we can call it a sphincter because yes, has it is angulated to the stomach as it inserts to the stomach. It actually creates a sphincter. Then the 29th was a question of genetic, basically. The only information that we did have to keep in mind is that the coloring blindness, it's something that is linked to the X chromosome, so it's a X linked recessive pathology. So we do know that Pietro did have this kind of genotype because he has coloring blindness, while Francesca, she does not have a phenotype of coloring blindness because she has two X's. So one of it was affected by the mutated gene, but the other one not. So whenever we want to know about the childhood that they have, we do have this. We do have to do this kind of Punnett square here, and whenever we consider the female child, so the daughters of Pietro and Francesca, we can only consider that line that I was pointing out, the blue one. And so the probability of having an affected daughter is 1/2, because we do have half of the probability that it is a female, and 1/4 of the probability in general on all the sons that they may have, all the child that they may have. So the correct option was C. So once again, remember, this is important, that whenever you have to find out the affected or not affected population with a X-linked pathology, whenever you do the Punnett square, well, there you only have to consider, if you only have to consider daughter or son, so female or male individual, you have to only count the lines where the you have actually the female or the male individual. Okay, so we do not care about the Y chromosome here because they were asking about the daughter. Okay, last three questions. So this one here asked us about how many hydrogenous bonds we find in this molecule. We have to remember that between the A and the T, we have 2 hydrogen bonds, while between the D and the C, we have 3. So starting from here, we have to count the pairs for each pair that we have. Sorry for the little tricky thing, and you have to multiply everything times 3 or 2, depending if it's C and G or A and T, and then we find out that it's 22 bonds. So basically the correct option was the E. So the 31, which was asking us what is needed for glycolysis to take place. We have to remember that we do have 10 steps in this process, 5 called endoergonic reaction, because in order to start the glycolysis, we need to give 2 ATPs, as you can see from the picture here, one here and one here. And so the only option that was correct, of course, was D, so ATP, while all the other ones were products of the glycolysis. And you have to remember as well that in order to start the process, we do need the coenzymes, but we do not need them reduced, we do need them oxidated, okay? So without the H that you see here, because whenever we have the H, it means that it's reduced and is the final product of the process itself. Okay, so the correct option was D. To move on to the last question, which of these statements is correct about the liver? They might be a little tricky because we are used to know that the liver is the biggest organ of our body, which is partially true because it is the biggest of our inner body, let's say, but the biggest organ of our body is skin, which is also the largest organ of our body. So the only option correct was that it can be called an amphicrine gland, because it both produces some secretes that are, for example, the biles that we have in the bile ducts here, and some hormones and certain proteins that are secreted into the bloodstream. So it has both endocrine and exocrine function, and so we call it an amphicrine. Okay, so the correct option was letter D. Remember, the skin is the heaviest and the largest organ of our body. So I stopped the share for biology. As Sarah said, if you have any question, just ask us on the chat and we will answer.
Okay, so now we're gonna see the chemistry part. Can you guys see the presentation here?
Yes, but now it's charging. Okay. Yes, now we are.
Okay. Okay. So let's start with the first question. So we are given.
Wait a second, it's a little zoom, and you can you zoom out a little bit the presentation, because I think you
Like this.
yes, like this is maybe perfect. Try
Okay.
to share.
Okay. So the first question asks us to find the yield, the theoretical yield of methane. So the total grams, total mass of this reaction. And the reagents of the reactions are aluminum carbide and water. And they react to give off methane. So the first thing that we need to do is to balance our reaction, that it's already written in your slides. So we balance the reaction, and usually when you have to balance reactions, you gotta start from metals. So in this case, aluminum, then you balance carbon, and then in the end, you balance hydrogen and oxygen. So after we've balanced the reactions, the first thing that you need to do in stoichiometry exercises is to find the moles. So whenever you feel stuck, you gotta calculate the moles. So we calculate the moles of aluminum carbide using the formula of moles. So we're going to have to divide the mass in grams of the aluminum carbide by the molar mass. And how do we calculate the molar mass? We simply have to add the molar masses of the single elements that create the molecule. So we're going to have to divide 125 grams by 144 grams times moles, and we find the total moles of aluminum carbide, which are 0.87 moles. At this point, we have to set up a proportion. So we have to set up the proportion between what? Between the moles and the stoichiometric coefficient of aluminum carbide and the stoichiometric coefficient of the methane, because what we need to find in this case are the moles of methane, which are 2.6 moles. In the end, we only need to find the mass, so the theoretical yield of this reaction of the methane, and we simply have to invert the formula of the moles. So we need to multiply the moles, so 2.6 times the molar mass of the methane, and the final answer is 41.6 grams. So the correct answer is C. Moving on to the 34th exercise, we are asked to consider these 4 molecules and find which of them has 2 pi bonds. Okay, so in order to solve this exercise, we have to keep in mind how many pi and how many sigma bonds are found in every type of bond. So the single bond usually has 1 sigma bond. The double bond has 1 sigma bond and 1 pi bond, whereas the triple bond has 2 pi bonds and 1 single bond. So in order to solve this exercise, we have to basically draw our molecules and see which type of bonds are contained within the molecules. So in the CH4, so in the methane, we have 4 single bonds. So we're going to have 4 sigma bonds. In the CO2, so in the carbon dioxide, we have the central carbon, which is linked to 2 atoms of oxygen through 2 double bonds. So we're going to have 2 sigma bonds and 2 pi bonds. Whereas the ammonium, which is a cation, has 4 single bonds, as in the case of methane. So we're going to have 4 sigma bonds. And then the hydrocyanic acid, which has the central carbon, which is linked to the hydrogen through a single bond, so 1 sigma bond, and to the nitrogen on the other part through a triple bond. So we're going to have 2 sigma bonds, 2 pi bonds, and 1 additional sigma bonds. So in the end, we're going to have 2 pi bonds and 2 sigma bonds. So the molecules that actually have 2 pi bonds are CO2 and hydrocyanic acid. So the correct answer is E. So the 35 is an exercise about the colligative properties, more specifically about the cryoscopic lowering. And we're asked here to see which of these molecules has the highest freezing point. So this is the formula here of the cryoscopic lowering. In this case, we need to calculate every cryoscopic lowering for every molecules, but we can neglect this coefficient because we are not given every coefficient for every molecules, and you do not need to know them for the IMAT. So in this case, we're going to only take into account the molality and the Van 't Hoff coefficient. We are already given the molalities of every molecule, so we're just going to have to calculate the Van 't Hoff coefficient. So we calculate the Van 't Hoff coefficient for every molecule, starting from the first one, and we find out that this for the first one, it is 3. For the second one, so the nitric acid, it's 2, because we have 2 species in solution. For the calcium hydroxide, it's 3. And for the phosphoric acid is 4. At this point, we multiply the molality times the Van 't Hoff coefficient that we just calculated, and these are the final values that correspond to the cryoscopic lowering for every molecule. At this point, we are asked to find the molecules with the highest freezing point, but watch out because actually, the relation between the freezing point and the cryoscopic lowering is an inverse relationship because the highest freezing point, the lower the cryoscopic lowering. So we're gonna have to select between our final results the molecules with the lowest DT. Therefore, the correct answer is A. So phosphoric acid and calcium hydroxide. Moving on to the 36, we are given some molecules here, and we have to select which are the acids, the bases, and the amphoteric. So in order to solve this exercise, we have to take into consideration these 3 definitions. So the definition of acids, bases, and of amphoteric species. So the basic species are the ones that actually in solution are able to accept a proton, so 1 atom of hydrogen. On the other hand, acidic species are the ones that are able to give off in solution a proton, whereas the amphoteric species are the ones that are able to do both, so both accept and release protons. And usually to identify the amphoteric species, we have to look for 2 specific factors. The first one is a negative sign. So usually amphoteric species are ions and specifically anions. So ions with a negative charge. And also you have to look for at least 1 atom of hydrogen in the whole molecular formula. So given this kind of inputs, we can actually select between our molecules here. So the first one is potassium hydroxide. So it is a base. Why? Because it is able to accept a proton. The second one, as you can see, is an ion with a negative charge and with 2 atoms of hydrogen. So in this case, it's going to be amphoteric. The first one is beryllium hydroxide. And as in the first case, it is a base. The third one, again, is another hydroxide, so again, a base. The fourth one is HCl, also called hydrochloric acid. So as the name states, it is an acid. Why? Because it is able to give off in solution an atom of hydrogen. The last one is again an ion, specifically with a negative charge and one atom of hydrogen. So it is able to both accept, but also able to release an atom of hydrogen. So it's going to be an amphoteric. So in the end, we are left with one acid, so the hydrochloric acid, three hydroxides, so three bases, and two amphoteric species. So the correct answer is E. 37 again is a stoichiometry exercises. So as you can see, they are pretty frequent in the IMAT. So if you are not really good at them, I would recommend to really try to hustle and try to master them. So we are given a reaction that we actually need to write between hydrogen and nitrogen that gives off ammonia. So first thing that we need to do, and we have to see how much ammonia, in this case, how many moles of ammonia are produced. So first thing that we need to do is to write the reaction. So we have 2 atoms of nitrogen plus 2 atoms of hydrogen, and we have the final ammonia here. Then we need to balance the reactions. So we start from nitrogen and then we balance hydrogen. And then what do we need to do at this point? Given the reaction, we need to find the limiting reagent. The limiting reagent is really important because it is literally the one that is limiting our reaction, because it is the first reagent between the reagents that gets saturated, so that finishes. And if we do not have any more a reagent, the reaction cannot keep on. So we need to actually find the limiting reagent in order to then calculate the moles of the ammonia. How do we find the limiting reagent? We basically need to get the moles, and in this case we are already given the moles by the exercise, and then we need to divide the number of the moles by the stoichiometric coefficient of that specific element. We have both because we have the moles, and then we have also the coefficient here because we already balanced the reaction. And we see that the limiting reagent in this case is hydrogen. Why? Because the limiting reagent has the lowest value, okay? So given the limiting reagent, at this point, we can set up our proportion between what? Between the moles and the stoichiometric coefficients. Keep in mind that this 0.17 is not does not have to taken into account. So once we find the limiting reagent, we have to completely delete and forget about these two values. So 0.2 and 0.17, because the moles, the actual moles are 0.2 and 0.5. So we set up a proportion, and the final result is 0.33 moles. So the correct answer is B. Okay, then we move on with 38. We need in this case to find the correct oxidation of our molecules here. So the first molecule is hydrogen peroxide. So keep in mind and try to remember that usually in peroxides, hydrogen has the oxidation number of -1. The second one is molecules in which we find fluorine. And fluorine is more electronegative than oxygen. So in this case, fluorine is going to have a negative charge and oxygen a positive one. So it's going to have +1. O2 is a molecular oxygen, which is an elementary molecule, so a neutral molecule. So the oxidation number is 0. And then we have the nitric acid, which has in which the oxygen has an oxidation number of -2. So the correct answer is B. We have then 39, which is which asks us to find which of these molecules have covalent bonds. We have 3 types of covalent bonds, which are polar, non-polar, and coordinate. In this case, the only one are the number 2, 4, and 5, because actually the lithium fluoride and the calcium hydroxide do not have a covalent bond, but an ionic bond that usually occurs between a really electronegative element and a non-so electronegative element like the alkali metals. So the correct answer here is C. 40 is again an exercise about stoichiometry, and it asks us to find the final mass of the ammonia. In this case, we have to first find the moles of our reagent, and we can do that by using the moles formula. So we need to divide the grams of our reagent by the molar masses, and we find that the moles are 2 and 4.5. Then we have to find the limiting reagent by doing the same thing that we did before. We find out that the limiting reagent in this case is hydrogen, and so we use the moles of hydrogen to set up our proportion. So we find actually the moles of ammonia, and then we multiply the moles of ammonia by the molar mass, and we find 51 grams. We're not over yet. Why? Because we need to find the actual yield. So the yield in this case is 80%. So we are going to have to set up another proportion in order to find which is the 80% of 51, which is actually 4.8. So the correct answer is D. 41 is about is a redox reaction, and it asks us to find the oxidizing and reducing agents. Oxidizing agents are the ones that actually gets reduced, whereas the reducing are the ones that gets oxidized. In this case, it is a really atypical reaction because it is at this mutation, meaning that we have an element that gets both reduced and oxidized. In this case, the element is chloride. Why? Because as you can see from the reaction, it both gets oxidized, so from 0 to +5, and from 0 to -1 in the reagents, in the products. Therefore, the correct answer is D. 42 is an exercise about the reactivity, and it asks us to determine which of the following is explains the highly reactive features of alkali metals when there is an increment in the atomic number. So when we increment the atomic number, what we're basically doing is adding more electrons to our element, meaning that we're going to have to add orbitals all around the nucleus. If we add orbitals around the nucleus, what is that supposed to mean? It basically means that the electrons are going to be further away from the nucleus. So the nucleus is will basically attract the electrons with lower force, lesser force. Therefore, the electrons are going to be more free to create bonds with other elements, with other atoms. And therefore, in chemistry, when we basically talk about reactivity, it basically means that elements and atoms are free to form bonds with other atoms. So if we increase the electrons, actually the element, the atom, is going to be able to find more bonds. So it's going to be more reactive. So the correct answer is B. 43. Again, stoichiometry exercise. In this case, we have to find the masses of our reagents, and we are given the final mass of our product, which are CO, so carbon monoxide, and iron. In this case, we have to find first we have to balance the reaction, of course, and then we need to calculate the moles of iron. We can do that because we are given the mass, and we need to calculate the molar mass. We find the moles, so we have 1 mole of iron. Later, we have to set our two separate proportions between iron and carbon, and iron and ferric oxide. So we start with the ferric oxide. So we calculate the moles of ferric oxide, which are 0.5 moles, and then we multiply it by the molar mass, and we find that we have 80 grams of ferric oxide. We do the same thing with carbon. So we find first the moles, and then we multiply the moles by the molar mass, and we find out that we have 18 grams of carbon. So the correct answer is D. 44 is an exercise about organic chemistry, and it asks us to see which of these molecules is a tertiary alcohol. So what is a tertiary alcohol? Tertiary alcohol is an alcohol in which the central carbon is basically linked to, on one side, an hydroxyl group, so an OH, and on the other three sides to three other atoms of carbon. So we look our answers here, and we have ethanol, which is actually a primary alcohol. So the central carbon is linked only to another atom of carbon. 2-butanol is a secondary alcohol. Propanol is a primary alcohol, and the 1-pentanol is again a primary alcohol. So the only correct answer is C. So 2-methyl, 2-propanol, because the central carbon is actually linked to 3 atoms of carbon. So correct answer is C. 45 is again an exercise about redox, and it asks to see which of these statements are correct. So in this case, we have to look to basically 2 elements in this case, silver and copper. We see that silver actually gets reduced because we start from a positive, so +1 oxidation number, and in the reagent we have 0. So it means that it loses it acquires 1 electron. Whereas the copper starts off from 0, so neutral state, and it goes to +2. So it basically loses 2 electrons. So silver is going to be the oxidizing element, and the copper is going to be the reducing one because it gets oxidized. So another thing that you have to look into is the fact that copper basically loses its 2 electrons, right? And where are these 2 electrons going? They're going to be transferred to silver. So in this case, the only correct statement between these 4 element statements is the second. The 2 electrons are transferred from copper to silver. So the right answer is B. 46 is the last stoichiometry exercises, and it basically asks us to find how many grams of white phosphorus are produced by this reaction. First thing to do, again, balance the reaction. Then, second thing to do is to find the moles. How do we do that? We set up our equation, our proportion between the moles and the stoichiometry coefficients. We find that we have, in the end, 0.3 moles of phosphorus, and then we use the moles formula, and we invert it by multiplying the number of moles times the molar mass, and we find that there are actually 36 grams of phosphorus. So correct answer is A. Last, for chemistry, is about organic chemistry again, and it asks us which of the following are the functional groups in asparagine. So first thing that you have to keep in mind is that asparagine is an amino acid, a non-essential amino acid, and as you have seen, I think, in biology, amino acids have really specific structures, and we have to, and usually when we refer to amino acids, we have to find 4 functional groups. One is an acarboxylic group, one is an amide group, and then we have an atom of hydrogen and the R group. So between our answers, we have to look for these 2 elements, amide and carboxylic. So these are to be, no matter what, present in our asparagine structure. Then we have to look at the molecules. So we have the amide here, we have the carboxylic, and we have another one. We have the amine. So the R group is represented by the amine functional group in this case, so H2N. So we're going to have in the asparagine an amide, a carboxylic, and an amine functional group. So the correct answer is D. Okay, so we're done with chemistry.
Okay, now it's time for math and physics. I will share my screen. Okay. So we start with the first question. Just one sec. I don't know why. Okay. Is it working now?
Yes, I can see the lights.
Okay, perfect. So the first question was asking us to find the correct option from the one below, and they were all property of the logarithm. So we need to remind which are the fundamental property. They are here displayed in the slide. It is 5. And typically, the only correct answer was the answer 1, which is the property of the logarithm of a product. And you can see here which is the rule. Then other important rule to remember are the logarithm of a quotient, the exponent rule, the change of base, and the inversion. So here the correct answer was A. Question 49, this was asking us, given this function, which is a parable, how can we make this a function bijective? So the parable we know is not a bijective function because it is a surjective. To have a bijective function, we can have, for example, a straight line. So among the options that they were given, the only one that was given as a straight line when arithmetically summed to the parable was the option B, because by simplifying the x square, basically we obtain a straight line. So this was it was the empirical method. We had to try all of them out, and in the end, the only possibility was the second. So correct answer is B. Then third exercise, it was giving us some definitions about median, mean, and mode. And so it was just a theoretical exercise. We need to know the definition. The median is the element in the middle of the data set, so the first option was not true. The mean and the median are not the same thing, so also the third option wasn't true. This actually is true, but only when the distribution is Gaussian, so in this case was not specified. Then the fourth option was telling us that the mean and the median are not the same thing again, so also this one wasn't true. And the second one instead is telling us that the mode is the element which is remitted the most in a data set, and this is the only correct option. So the correct answer was E. 51 was just asking us which polyhedron has a 6 phase among the different options. The options were hexagonal prism, which has 8 phases, so no. Pentagonal pyramid, which is actually the correct option because it has 6 phases. Triangular prism, which has 5 phases, so again is not correct. Hexagonal pyramid, which has 7 phases, so no. And finally, pentagonal prism, which has 7 phases. So correct option was B. 52 was asking us, giving a given number set, find the mode and the median. The mode, we said before, is the most common number, so the number which is repeated more time, and given this number data set, we can actually see that the mode was 2. And then we have to find the median. The median is the number in the middle of the number set, and in this case was 8. So the correct answer is C. Then exercise number 53, which is the last one for mathematics, this was asking us to calculate the compound function h that is given by g compound with f. When we have a compound function, what we have to do is that in the moment in which f has to be inside g, we have to rewrite first our g, and instead of the x, we have to substitute with the f function. So the compound function is e to the 1 over x. So the correct option was E. If you have any question, you can ask in the YouTube chat. Otherwise, we can pass to physics. The first physics exercise was asking us to calculate the weight of this body out of the water, given that we know the weight of the body when is immersed into water for 80% of its volume. To solve this exercise, we need to calculate the volume of the cylinder, and we can do it because all the data are given in the text. Then we can calculate the Archimedes force, and after it, we can calculate the weight of the body out of the water as the sum of the force in the water plus the weight in water. So the correct answer was the 226 Newton. Number 55, it was a thermodynamic exercise and is asking us the total heat needed for the water to completely evaporate. So we first need to calculate the heat that is needed to bring the water to the boiling point, and then we need the heat for the transition of state to occur from liquid to gas. So we first calculate the water to bring the heat the water to 180 degrees Celsius, which is the boiling temperature, and after we calculate the heat to boil the water, which is therefore the heat for the transition of state, and then we have to sum them. So the final answer was D, 6.85. Number 56 was about the electric field. So it was giving us different options, asking us to see which one is true. The first one is about the formula to calculate the electric field, which is actually given by the product between the Coulomb force and the charge. So the one given in the options wasn't true. The second was actually true because it was telling, it wasn't true because we know that the electric field can be generated by one electric charge, but also by multiple charge. So it wasn't true as well. And the third one, it tells us that the unit of measure is Newton over Coulomb or volt over meters, and that is actually is the only correct one. So the correct answer was D. 57 is about an iron ball which has a given mass, which we know, and we know that is immersed into water for this volume. What we have to calculate is which is the what percentage of the body is immersed. To do that, we have to calculate the Archimedes force, which we can do with the given data. And when we calculate it, we can see actually the weight of the ball is greater. This means that there is no percentage which is out of the water because the whole body will be totally submerged, so underwater. And therefore, the correct answer is in none of the above. 58 is about projectile motion. So what we have to calculate is the range of the cruise sheet, which the exercise tell us is as a certain velocity and is as a certain angle with the horizontal line. So knowing the angle, we can calculate the sinus and the cosine, and we know the initial velocity, so it's just applying this formula. And the correct answer is B. Then 59 is about, again, a theoretical question about the heat machine. Normally, by definition, a heat machine will convert heat into work. And all the given example of the options were true, except the except the electric heater, because the electric heater is not converting heating into work, but is using a resistor to produce heat. So it's a different way of, it's a different mechanism at the base. Last one is about the Ohm's law. So again, electrostatic. In this case, is asking us to calculate the current, given that we know the electric potential and we know the length. So what we can use is first the second Ohm laws, which allow us to calculate the resistance, given that we know the resistivity, the length, and the circular section of the wire of the wire, yes. And finally, we can calculate the current with the first Ohm laws. And the correct answer was C.
All right, thank you. So I'll share the final slides. So for you guys, once again, you have the evaluation questionnaire that we ask you to fill because it's very important for us. It will give us very important feedback for this kind of events. As you have seen, I already shared the link in the chat as well if you guys prefer. Then we keep you in mind to follow us on our social medias, such as on Instagram, on YouTube, with this channel we are taking the event today, and on TikTok as well. Because we are making some TikToks with question and other tips that you might find interesting to know. Finally, thank you for coming. We're giving you this discount code, which is 15% off of all our starting courses, and it's valid until the August 19th. The code is the one you find here, so MS_event_1208, all in caps lock. So I think this is the end. So goodbye and thank you for watching. I'll let the other girls to say goodbye to you as well, and feel free to leave whenever you want.
Thanks for joining and have a good evening.
Yes, thank you and good luck with the test.
Thanks, good luck, bye.
Bye
Bye,
bye.
guys.